Hey everyone, welcome to the 7th edition of our ActinoBase e-seminar. Today we will be having talks from Bernhard Kepplinger, postdoc from the University of Newcastle, and he's going to be talking to us about the 3 horsemen of the Amycolatopsis. And then after that we'll have Patrick Capel, or Pat. He's a PhD student from the University of Warwick, and he's going to be talking to us about cell-free transcription translation for natural product research. Make sure you hang around to the end. I've got some exciting stuff to talk to you about the future, excuse me, of the ActinoBase e-seminar series, including a special we'll be doing in replacement kind of the ISBA meeting later this month. So first up is Bernie. So take it away, Bernie.
Sorry, Bernie, you muted just now.
Sorry about that. First of all, thank you very much for the organizers to put this seminar series together. Today, I would like to show you some work I've done doing my PhD and later on finished off. So that, most of the stuff was done in collaboration with the Mirrors, which is a small spin-off company. And I'm going to talk about the 3 compounds we discovered from this very talented Amycolatopsis strain. So first of all, just to give you some background, I think most of the audience probably know this. Most of the antibiotics were discovered in the so-called golden era of antibiotic discovery. And afterwards, not many of the new classes were actually discovered. In addition to this, the resistance to a lot of these compounds developed very shortly after this. And this is creating a real problem these days. So where do we take new antibiotics? As probably most of the audience know, actinomycetes are this really great source of new compounds, specialized metabolites. They are filamentous soil bacteria, and they have a genome a lot larger than those of Bacillus or E. coli. And they're producers of valuable compounds, not just antibiotic, but anti-cancer compounds, immune suppressants, and so on. And they are actually also the starting point for most of the clinically used antibiotics. So a lot of them were afterwards modified for additional purposes. And they also have this beautiful life cycle, and they grow in this filamentous fashion from spores. And they also look stunning on the plate. And they also smell a lot nicer than other bacteria, if I'm allowed to say. One of the model organisms, Streptomyces silical, it's able to produce at least 3 compounds, which we knew about, but it came as a real surprise when they sequenced the genome that it's actually capable of producing a lot more compounds than actually just those 3. And this opened up really a new era of antibiotic discovery, this so-called genome mining technologies. The Mirrors, which is a small spin-off company, works from the collection of Michael Goodfellow. Those of you who don't know Professor Goodfellow, he's a taxonomist, and over a long-standing career, he really tried to discover new species from a very diverse area. So this library is actually unrepresentative in the sort of content and dereplication, which a lot of other Actinomycetes libraries suffer from. And the strain I'm going to talk to you about today is the strain DM30355. I'm just going to call it 355. And it was actually isolated from the Atacama Desert, which is supposedly one of the driest places on Earth. And the idea behind this, if you look in new environmental niches, which nobody else looked before, you might find new chemistry, which was indeed the case in this type. My former supervisor, Professor Dr. Allenby, got a small grant to ferment some of the strains from the Goodfellow collection at the CPI. The CPI, Center for Process Innovation, is a facility where you can actually do large-scale fermentation. And we fermented this strain at the 500-liter scale, and I designed a downstream procedure to purify this 235 grams, for better word, brown sludge. When I separated this further, we saw actually multiple compounds are present in there. And this is a lipid 2 reporter strain, so it goes blue in response to certain stresses, in this case, cell envelope stress. And one of the most hydrophobic compounds is this compound, which we named tatiomicin. Unfortunately, I'm not allowed to show you the full structure of those. We hope to put the patent in for this week. A bit late for this talk, but that's what it is. Another compound I want to talk to you about it is this ansamycin type antibiotic, clindamycin A. And the last compound is called Vancoresmycin. And we also sequenced the genome of this strain, and it's in size similar to other Amycolatopsis strains. And we assembled it with the help of PacBio data and Illumina data into 13 contexts. And we actually found the gene cluster responsible for each of those. And I'm going to talk about it a bit more in detail later on. The first compound I want to talk to you about is this novel polyketide, which we're currently in preparation of publishing. So when we looked into the genome for this Amycolatopsis strain, we found a cluster which, based on the structure, could be responsible for tatiomicin production. We put the cluster through BigScape, and we indeed saw that it clusters closely to rishirilides and alnomycin. However, the gene synteny is really altered compared to those. And if you look a bit into more detail, the gene synteny is completely different, and we found additional genes which might be responsible for this novel structure. However, we needed to prove that this gene cluster is really responsible for tatiomicin production. So I transferred the whole cluster into the surrogate host Streptomyces silicala M1152, which is largely void of antibiotic gene activity and is one of the so-called superhosts. And you can see that we found additional peaks. And the peak here in gray corresponded with a standard purified from the native host. And when we look at the mass spec of these compounds are indeed identical. So we were able to prove that this gene cluster is indeed responsible for tatiomicin production. So why we're interested in this compound? It is active against a broad range of gram-positive pathogenic isolates. However, largely, but it is not active against gram-negative or yeast. However, when we looked a bit more in detail in it, it is, as you can see here, completely inactive against E. coli. But when you use an E. coli strain which lacks the outer membrane, it is active again, which means the antibiotic target is probably still negative in gram-negative organisms. However, it is not taken up into the cell. However, what is the mode of action of this compound? We used we used reporter strains where the promoter for the reporter gene is fused to a laxat gene, so they go blue in response to those antibiotics. However, when we tested it against a panel of those, our compound was not giving any signal, which wasn't really helpful, but might indicate that we have a novel mode of action for this compound. So there are many ways of how to distinguish which targets could be responsible for what could be the target for tatiomicin. We chose in the next step to do a cell morphology assay. So this is just Bacillus subtilis, happily growing without any compound. And you can see we tagged the chromosome with NFP, which is fused to a histone-like protein. And we also target the membrane with an artificial membrane domain. And this one is happy growing. However, if you give, for instance, niacin, which is a pore-forming antibiotic, you can see the membrane is getting spotty. On the other side, if you use rifampicin, which is an RNA polymerase inhibitor, the chromosome completely decondenses, which is very characteristic for RNA polymerase inhibitors. And when we now did the same for our new compound, tatiomicin, we could see the same chromosome decondensation. So this might mean that tatiomicin is indeed targeting the RNA polymerase. However, we need to still do a lot more further investigations to prove this. The next compound I want to talk about is this linear polyketide NABS hybrids molecule, which is called Vancoresmycin. Vancoresmycin was originally discovered in 2002, and it has this linear shape. It's linear polyketide with this tationic acid warhead, and it is very active against gram-positive organisms down to the nanogram per milliliter scale, so a very potent compound. However, the mode of action for it was unknown. Again, we used a reporter assay, in this case, a cell envelope reporter, and as you can see, Vancoresmycin really nicely activates it, and this gave us a good hint that this might be the mode of action. However, certain cell wall compounds, and vancomycin, for instance, also targets also activates this compound. So in the next step, we did a depolarization assay to check whether or not the compound depolarizes the bacterial cell membrane, and we used a voltage-sensitive dye disc for this. And as you can see, Vancoresmycin is indeed depolarizing the bacterial cell membrane in a dose-dependent manner. We can also check this under the microscope. So if the cell is nicely polarized, you can see this nice disc signal. However, when you depolarize it with, for instance, gramicidin, which is a small ion channel compound, the fluorescence vanishes. The same for niacin, which is, as already said, a pore-forming compound, which also depolarizes the membrane. We then coupled this with another dye, which is called cytoplasmic, which can only enter the cells through large pores. And as you can see, niacin, which forms these large pores, allows the dye to enter and stay in the chromosome. However, gramicidin, which forms small ion channels, does not. And Vancoresmycin indeed depolarized the membrane in a non-pore forming fashion. And we can also quantify this. In addition to discovering the mode of action for Vancoresmycin, we transferred the entire 141 kb gene cluster from Amycolatopsis 30355 into Streptomyces silica M1152. And these are some of the X-conjugates, which then go nicely blue on this lipid 2 reporter strain. And we got, again, a peak corresponding to the Vancoresmycin standard, and the mass was identical. So we're very happy that this is the antibiotic gene cluster. However, how does this help us? The gene cluster for Vancoresmycin was previously unknown, but based on and also the stereochemistry of this compound was unknown. And based on the domain analysis of the dehydratase, ketoreductase, and alkyl transferase, we were able to predict the stereochemistry of this compound, which I think the natural product chemists would appreciate would be incredibly difficult with such a flexible molecule otherwise. The last compound I would like to talk to you today about is Canglamycin A, which is an ansamycin derivative. Canglamycin A is not a new compound. It was actually discovered in 1988, so a very old compound. So why were we actually interested in this compound? It shares great similarity to rifamycin S, but what we found already very early on that is active against rifampicin-resistant strains, and we were really curious about how it actually does this. We tested it against a variety. This was in collaboration done with Professor Senkin's group, especially Hamed Mossai, who was spearheading this research. And as you can see in the wild type, both compounds have a similar IC50. However, when you mutate the active center of the RNA polymerase, you can see that rifampicin loses its activity. However, clindamycin A largely remains activity against the RNA polymerase. We then collaborated with the Muramaki group at Penn State, who have crystals of RNA polymerase, and we soaked the compound into these crystals. And you can see here that the structures are indeed very similar. We could show that clindamycin A is forming additional bonds to the RNA polymerase and can therefore overcome this resistance. And we also tested it against Mycobacterium tuberculosis. As many might know, rifampicin is the first in-line treatment for TB. And we tested it against a multi-drug resistant strain of TB. And as you can see, clindamycin largely remains active against this multi-drug resistant isolate of TB. Although the MIC is a bit higher in general against rifampicin and rifampicin, these are heavily modified structures. They have this additional piperazine group, which has additional properties for the compounds to allow the bacterial cells. And this is why we believe the compound isn't quite as active against wild type as, for instance, rifampicin or rifampicin. So to summarize up, I'm sorry, I was speaking quite fast actually. Actinomycetes continue to be a valuable source of new and repurposed antibiotics. The Amycolatopsis isolate 30345 produces at least 3 antibiotics of separate classes. Tatiomicin is a novel tricyclic polyketide with a so far undetermined mode of action. However, we believe that it might interact with the RNA polymerase. Vancoresmycin is a potent antibiotic which acts via membrane depolarization. We were able to transfer the entire 141 kb gene cluster into a surrogate host and to predict the stereochemistry. And clindamycin A is a rediscovered compound that is active against rifampicin resistant tuberculosis, and further studies into its clinical applications are currently in progress. I would quite like to thank all the collaborators and my former supervisor, Dr. Allenby, and my current supervisor, Professor Arrington. I would like to thank the whole Senkin group, in particular Nikolai and Hamed, the Strahl group who collaborated with us on the determination of the mode of action of Vancoresmycin, the Muramaki group for doing the soaking of the clindamycin A into RNA polymerase, Michael Hall and his team for a lot of structural determination, and the Perry group in the Fremont Hospital for determining the MSC against clinical isolates. I'm happy to take any questions. Thank you very much.
Well, thanks, Bernie. So yeah, as you know, you can put some questions in the YouTube chat. Well, we've got one now, and while we wait for a few more to filter through. The first one is from Nerds410. He asks, what method or methods did you use to clone the 141 kilobase cluster for cloning and conjugating into the M1152?
We used a very standard method. We asked BioSNT to create a PSAC library, which is this bacteria artificial chromosome library. And from that, we then conjugated it into M152.
Okay, well. And then Anika asks, are these antibiotics secreted? Any transporter in the cluster responsible? And also is toxicity in mammalian cells for these? And have you checked the toxicity for mammalian cells for these antibiotics?
We have checked the toxicity for Vancoresmycin, which is indeed toxic. This is probably why it went never further into development, because it was actually discovered by a company. Toxicity studies with tatiomicin are still in progress. And for clindamycin A, it is actually non-toxic. That's why we are so interested in the further development. It's very similar in structure to the current classes, and therefore we saw no difference in toxicity there. Cool. But that's also preliminary data, I should say. Yeah.
So, I mean...
the other question was about the transport of those.
Yeah, yeah.
In tatiomicin, we see multiple transporter. All of these compounds are secreted. which is normal for antibiotics. We don't know which transporter is exactly responsible for tatiomicin. We also don't know if some of those transporters might be responsible for uptake, because all you can really say it's a transporter. So there's a lot more to still be done with those compounds.
Cool. So this Amycolatopsis strain, is it very genetically tractable? Have you done kind of genetics work with it in vivo, or has most of it been transferring it over to the superhost?
We haven't done any real, how should I say, we didn't do anything with the strain itself. We mainly transferred it, and there we only really did heterologous expression. We didn't target the clusters yet themselves. Okay,
so you've had no issues with expression. You've just chucked them into Color and they've expressed.
I mean, heterologous expression is always a bit black magic and luck. I mean, everybody who tries this will agree with me there, I would say. However, we were able, after a while, to get it to work, but we didn't really have to alter the conjugation procedure. We mainly think that the conjugation efficiency has to do with how large your bacterial artificial chromosome library is, because, for instance, if you use a cosmid library, the efficiency is generally higher. It is also works better if you don't use the methylation deficient strain ET. We think that this strain might be responsible for modifying some of the clusters itself. However, it also increases conjugation efficiency. So we don't know. Sometimes you get the cluster in, but it's not active and you don't know why. That might be a reason for that.
So what are you using instead of ET?
We use ET at the moment, but when we use for Silicolor, we have to. But for Albus, you don't have to use the ET strain, for instance, because it lacks the restriction enzymes for it.
Sure. Devas, have you identified any resistance mechanisms to the molecules?
For Vancoresmycin, we didn't look because we had such a good we had such a good reporter for it. Also, we were not interested in further development. For Canclamycin A, you need 2 separate mutations in order to create resistance, which is different to the rifampicin molecules. However, they then have a real fitness costs for the RNA polymerase. Yeah. And for tatiomicin, I tried very hard to get resistant mutants. I was not able to, and I still don't know why.
Ah, that's really nice. Okay. If anyone else has got any more questions, remember you can put them in the chat, or you can tweet hashtag ActinoBase 07, I think it is this time. And I'm sure Bernie can find some questions there and answer them as well if you have them. So thank you very much, Bernie. And next up, we have Patrick Capel from Warwick talking to us about cell-free transcription translation for natural products. So Pat, if you're ready, you can go ahead, share your screen. Awesome. Take it away, Pat.
Lovely. Thanks for the introduction, Tom. So my name is Patrick Capel. I'm based at the University of Warwick, where my primary supervisor is Amded Los Santos, and I'm also supervised by Christoph Kaur. I'm funded by SynBio CDT in partnership with Syngenta, where my main contact is Jason Vincent. And my PhD project is titled Cell-Free Transcription Translation for Natural Product Research. So what is cell-free expression? Well, cell-free expression is the reconstitution of transcription and translation in vitro. For those of you who may have been a while, of course, transcription is the process of creating mRNA from a DNA template, and translation is the process of creating a protein from an mRNA template. Very simply, transcription requires an RNA polymerase as well as dNTPs, and translation requires a ribosome as well as amino acyl tRNases. Now, if you wanted to reconstitute this in vitro, you need 3 things. You need some template DNA, you need the biological machinery you require, as well as all of the other small molecules that go into making this process work. So when you reconstitute this expression in vitro, you need your DNA, obviously. Most commonly, this is done in the form of plasmids. However, some people in various research groups also use linear protected DNA, where a piece of DNA is protected from exonucleases via proteins. Now, something to note here is that your final DNA must be very pure. If you have a lot of contaminants from salts from things like MIDI preps or maxi preps, your TXTL reaction, your transcription translation reaction will not work. And also, you need a lot of DNA. Just for some reference here, I use a final DNA concentration of 25 nanomolar of my plasmids in my reaction. So I spend a lot of my time doing a lot of maxi preps. You also need the biological machinery. Of course, you need your transcription and translation machinery, which includes your RNA polymerase and your ribosomes, but you also need other things such as sigma factors and chaperones to facilitate the correct folding of your protein. You also need a lot of machinery that's involved in recycling of nutrients inside of a normal cellular system, such as those involved in ATP synthesis. And this, of course, helps your cell-free reaction to continue for longer. And if you've isolated all of this machinery very crudely from an organism, you also have some of your reagents here, such as your amino acid tRNAses or your DNTPs. And this is affectionately known as your cell-free extract in the world of cell-free transcription translation. The final component I mentioned that you need is a master mix. And your master mix is all of those small molecules that are required for this to occur. And I'd like to break this master mix down to 4 main groups. The first group being your reagents, things that are explicitly used up in your cell-free reaction like DNTPs. The second being buffers, such as HEPs or PEG 8000, which is a molecular crowding agent. I like to think of it as a spatial buffer. You also need compounds that are involved in reduction, such as NAD, or oxygen-free radical capture, such as spermidine. And you also need some other small molecules that are involved in protein synthesis, such as folinic acid. Now, this master mix is the major cost of running a cell-free reaction. To get all of the components at the correct quality and purity, you're looking at about 1,100 to 1,300 pounds for 1,000 reactions. So it works out at just over £1 a reaction. So those are all the components you need for a cell-free reaction. But how do you actually run one? Well, very simply, you take your 3 components, your DNA, your extract, and your master mix, and you mix them together and aliquot them out into 3 technical repeats. I make a large batch of this mix. I make 33 microliters and spit that out into 3 10 microliter technical repeats. I then let those technical repeats incubate, whatever temperature is required for my particular reaction. And after 6 to 24 hours, I'll read out the output of my reaction. And the output could be the production of a fluorescence protein or potentially even the production of a small molecule that you could detect via mass spec. It's relatively straightforward, but it requires a lot of pipetting accuracy. So that's a very brief introduction to cell-free transcription translation, but now a little bit on natural products. So natural products are really interesting organic compounds that can be isolated from biological sources that are often the product of primary or secondary metabolism. I'm particularly interested in polyketides and non-ribosomal peptides. Polyketides, of course, being biosynthesized from acetyl building blocks, and no prizes for guessing that non-ribosomal peptides are synthesized from amino acids. Some examples of polyketides include deoxycycline and aflatoxin B1, deoxycycline being an important tetracycline antibiotic, and an example of a non-ribosomal peptide being enterobactin, a siderophore. Now, although these chemical structures are very beautiful, and these compounds or other compounds like them have many important applications from the agrochemical to the medicinal, their biosynthesis is also just as interesting. And they're often biosynthesized on a biosynthetic assembly line. And all of the genes required for such an assembly line are often found clustered in something which is known as a biosynthetic gene cluster, quite an imaginative name. At the core of this biosynthetic gene cluster is your PKS or your NRPS genes, and they synthesize the main scaffold of your compound you're interested in. You'll then also often find tailoring enzymes that decorate this scaffold, such as performing halogenation or hydroxylation reactions. If this compound is toxic, you may also find resistance genes to prevent the organism from suffering any of the negative effects of producing a toxic compound. And there are also regulation genes, which determine when this whole biosynthetic pathway is turned on or off. Now, thinking further about the NRPS or the PKS genes, these encode for things known as megasynthases. And these megasynthases can be broken up into modules and domains. So each megasynthase will be composed up of a number of modules, where each module is responsible for the complete incorporation of one building block. These modules can be further broken down into domains, each of which are in turn responsible for a single catalytic action. And something that's interesting to note is that your growing natural product chain is covalently tethered to your megasyntheses via phosphopantothene arm. This is a hypothetical example here, where the first module, an amino acid, is loaded, and subsequently 2 acetyl units are loaded and subsequently modified slightly. So that's it for natural products. But why would you want to work with cell-free in the context of natural products? Well, the first thing is you can go around every synthetic biologist's favorite cycle, the design, build, test cycle, very quickly with cell-free expression systems. As I mentioned earlier, one reaction, one expression, takes anywhere between 6 to 24 hours. When you compare this to the scales of heterologous expression, which could be weeks or even months if you're unlucky, you can go through design, build, test cycles incredibly quickly. In the context of natural products, then, you could do a lot of things. You could potentially engineer biophysical interaction between 2 megasyntheses that don't normally communicate with each other, producing a chimeric natural product. You could also do things like tailor tailoring enzymes to maybe make them more promiscuous or perform a slightly different function. And you could even play around with the catalytic activity of various domains in a megasynthase to maybe incorporate a different building block. And another interesting thing to note here is we have a decoupling from physiological constraint. As you have no living cells here, you don't need to worry about the production of any toxic compounds because you have no cells to kill, unless, of course, the compound you're producing interferes with transcription or translation. You could also do things like very quickly realize the real-world output of in silico discovered genes. Over time, we're getting better at discovering genes in silico and even predicting what their function will be, but you do need to verify that in the real world. With a cell-free system where you could order your DNA from your favorite DNA synthesis and drop it into a cell-free reaction, you could potentially go from discovery to real-world output in a number of days. And finally, the cell-free reaction is pretty much a chemical reaction. It's an open system. It's not a closed system like a living cell is, where you can drop in anything you want into your cell-free reaction. So say you hypothesize that your natural product pathway needs an exotic cofactor. You don't need to worry about creating a strain which can produce this cofactor or feeding a strain this cofactor, because you can just drop it simply into your reaction. It's a chemical reaction. So what organism am I extracting my biological machinery from? And my debate is Streptomyces versus E. coli. Seeing as I'm talking here the ActinoBase, you can pretty much guess that the answer here is Streptomyces. But E. coli, of course, is a very well-studied model organism with a very mature genetic toolkit. And there are even commercially available cell-free platforms that you can buy, but all you need to do is add DNA and you're off. However, E. coli has historically not been the best at expressing Streptomyces genes, or other Actinomyces genes for that matter, especially those that have incredibly high GC content and are long, such as the megasynthases. For this reason, I've chosen Streptomyces as the organism from which I'll isolate my biological machinery. So what are the goals for my PhD? Well, the first one is to make functional Streptomyces extract. The second goal is to create a genetic toolkit so I can control the levels of expression inside of my cell-free reaction. And my third goal is to prove the platform's usefulness in a natural product context. So the way I produce my extract is very straightforward, and I create a crude extract. I take my Streptomyces strain I'm interested in, and I grow it up and subculture it and culture it in liquid media. Once it's grown sufficiently, I harvest it via centrifugation, washing it with buffers that contain salts like magnesium and potassium, reducing agents such as DTT. I then lyse my cells via sonication in a minimum volume of buffer and centrifuge that very hard to hopefully remove any cellular debris I'm not interested in, such as membranes, and decanting off the top, which should contain all of my interesting biological machinery. Now, this is a fairly crude extract. Now, there is a question of complexity versus simplicity here. As I said earlier, when I run my cell-free reactions, I have three major components: my DNA, my master mix, and my cell-free extract. But in the cell-free sphere, however, people have been adding other things to their cell-free reactions to make them more efficient or to make them run for longer. And examples of these other components could be things such as exogenous tRNA or transcription and translation factors, such as the elongation factor or release factors. And in fact, this work has very recently been published for Streptomyces this April, where the addition of exogenous tRNases, as well as a specific combination of these other factors, provided an 8-fold increase in activity. Now, this extra activity is obviously greatly beneficial, but that does require every extract you produce to be optimized further with these other factors. So providing I can have a high enough level of activity of my extracts, I'm going to try and keep things simpler. So for my extracts, I've been able to produce extract from a couple of different Streptomyces strains, including Streptomyces venezuelae, albus, avamitolis, and lividans. And a thing to note here is whenever I say the activity of my extract, I'm basing this on the fluorescence. And I'm doing this because when I run my reactions, I'm running them with the DNA being a plasmid, which encodes for M scarlet. And I chose M scarlet over GFP because I think the hot pink's quite pretty. So as you can see here, all of these strains produce some amount of M scarlet. And interestingly enough, the addition of lysozyme or not adding lysozyme does have differing effects on each strain. For strains like Streptomyces venezuelae and Streptomyces albus, especially Streptomyces lividans, the addition of lysozyme during the lysis stage provides an additional activity level. However, for Streptomyces avamitolis, if you add lysozyme when you're lysing the cells, you see that your extract is completely dead. Unsure as to why this is the case now, but it was interesting to observe. As for the genetic toolkit side of things, why would you need a genetic toolkit in the context of a cell-free platform? Well, controlling the level of expression in a cell-free platform is incredibly important, especially when you start dealing with multi-protein or multi-protein systems. Being able to fine-tune the level of expression of each protein or each enzyme in a pathway can do things like prevent the production of shunt metabolites, or indeed force them. And my genetic toolkit is based around a standard plasmid, which I edit by adding the operator sequences of varying transcription repressors in. And the reason why I've chosen a transcriptional repressor approach rather than, say, a suite or a library of promoters and ribosome binding sites to offer different levels of expression is so that I can reduce the amount of cloning that is required when I'm dealing with larger libraries. On the side of the transcriptional repressors, I've chosen LAC-I and TET-R, two very commonly used transcriptional repressors. And I've also chosen MMFR, which is a transcriptional repressor that is found natively in the, I can't say this antibiotic's name now, is found natively in Streptomyces venezuelae. What I've done here is I've cloned in the operator sequences into the standard SP44 promoter, such that they will be recognized by these transcriptional repressors. And I have done that cloning. Now, when you have this new plasmid, which has been slightly modified, you'd hope that in a cell-free context, at least, it would still produce a large amount of the protein you're interested in. When you then run another reaction with exactly the same plasmid, which has been engineered, as well as the transcriptional repressor that is encoded for in the operator sequence is encoded for in the promoter, you would hope to see no expression or reduced expression depending on the concentration of your transcriptional repressor. The transcriptional repressor here being this little horseshoe cartoon. And finally, when you add your small molecule inducer along with your transcriptional repressor, you'd hope to be able to recover the level of expression that you had originally. Hypothetically, then, you could produce a 2D heat map with varying levels of your transcriptional repressor and your small molecule inducer, and therefore hopefully see varying levels of expression. Just to reiterate, when you have no concentration of your transcriptional repressor or your small molecule inducer, you'd hope to have a high level of fluorescence. Adding in your transcriptional repressor, you would hope to see a decrease in your level of output, in my case, fluorescence. Adding in your small molecule inducer, you'd hope to be able to recover the activity of your transcription translation reaction. And finally, you would also hope that your small molecule inducer does not have any meaningful impact on your cell-free reaction. And I have the data here for the LAC-I set, where I've used the LAC-I operator sequence SP44M scarlet plasmid. This is using a Streptomyces venezuelae extract, and I'm incubating overnight. And as you can see, the data pretty much fits the trends that you would hope to see, with high concentrations of LAC-I providing a very low level of fluorescence, adding IPTG to other reactions with your high level of LAC-I producing, well, recovering some level of your output, in this case, fluorescence. And finally, the addition of IPTG itself does not seem to have that greater effect on my cell-free reactions. Now, due to COVID, this is pretty much where I've got up to now. I've been able to make some functional Streptomyces extract, which is great if that's the idea of your whole PhD. I've also started creating a genetic toolkit to control the level of expression inside my reactions, and I'm working on proving the platform's usefulness. Some future work would be to flesh out the genetic toolkit, obviously, and also to optimize the extract production for all of the strains shown. This includes tuning levels of various different salts, as well as maybe adding in some commercially available additives, such as polyvinyl sulfonic acid, which is an RNase inhibitor. And I'm also working towards demonstrating the platform's usefulness with some natural product examples. So with that, I'd like to thank Enzo and Chris, my supervisors, as well as their respective lab groups for friendship and moral support. I'd also like to thank Syngenta and Jason for extra funding for my PhD, as well as the SynBio CDT for training and funding, and the University of Warwick. I'd also like to thank the organisers for the opportunity to speak and you all for your attention. And I would love to take any questions you may have.
Well, thanks for that, Pat. As always, just waiting for some questions to come through, but the first one is from Anika, who asks, how do you check the misfolding of any protein, and how different is the yield in a cell-free pathway?
Yeah, so if you're thinking about protein folding, in cell-free, what you could really do is you could either see if your protein is produced and it is the correct length or length you'd expect. And if you have misfolding, I would imagine you'd have to basically look to see if you're having any level of activity. And then if you're not seeing any activity, but you do see a protein that is the correct length, you could do other things to try and optimize your extract to make it fold better, maybe by adding in varying amounts of PEG or by adding in any chaperones that are known to help in that pathway.
Cool. Another question, Nerds410 asks, do you know if using the MMFR system can be used for in vivo inducible expression in Sven as well?
So I believe some work is being done on that in Christoph's lab group at the moment. But other than that, I don't know yet.
Cool. And then another one was, do you know if you can use your system to test translational inhibitors?
That's an interesting question. Translation inhibitors. So providing you had a way of positively showing you had transcription or translation, you could then add in whatever you wanted to test to see if translation was therefore curtailed by the addition of that product with the correct controls.
Yeah.
It'd just be a question of experimental design there.
Yeah. I'm really curious as to why you picked 48 hours for your cell-free extract for when you're taking that, as in, you know, how does that match up to the life cycle of the Streptomycetes you picked, and kind of like why you've chosen that time in the life cycle?
Yeah. So this is something we've spent a lot of time thinking about. So I try and harvest my cells at the end of the exponential phase, because that's when most other cell-free systems that the protocol for other organisms such as E. coli are always harvested at that particular stage, when you're trying to get it right at the end of the exponential phase. Now, of course, when you think about natural products may be produced at varying different stages in a Streptomyces life cycle, such as in a stationary phase. So far, I've picked the end of the exponential phase to try and catch what is commonly known as the better time to catch it. But we are thinking about maybe optimizing our extracts for the production of natural products by harvesting them later or earlier. Of course, this will also have lots of other effects, such as changing the concentration of different proteins that are involved in transcription and translation, and subsequently the maybe chaperones for correct folding. Yeah.
Okay, cool. So I think in the essence of time, if you have any more questions, please use Twitter and tweet at Pat I'm sure he'll answer those there. I'm just going to share this again and talk about what we've got coming up. So next week, Thursday, the 18th of June, Dr. Virginia Helena Alvarezin, and I'm sorry if I've murdered that surname, who's the head and group leader at the Center for Electron Microscopy at the National University of Tucumán, is the first PI giving a longer talk, so it'll be around 40 minutes, and she will be talking about UV-resistant actinobacteria from high altitude Andean lakes, the diversity of molecular mechanisms. So make sure you tune on for that. That's going to be later time at 7 p.m. BST on next Thursday. But also, we've been talking about what to do as ISBA's been cancelled this year, well, postponed till next year. And so on Thursday, the 25th of June, we're doing an ISBA special. We'll be releasing some more details about that later. But if you're interested, we're going to be doing a series of flash talks for early career microbiologists. These are going to be 5 minutes long and a 3-slide maximum. So we've tweeted about it. Make sure you go sign up for those talks. The more the merrier, and it would be great to have you for those talks. So without further ado, that's it for this week. Make sure you tune in next week. Thank you so much for joining us this week, and see you soon.